So in this session I'm going to cover some more targeted therapeutics as anti-cancer therapies, looking at proteasome inhibitors and epigenetic modifiers. There is a more complex version of the epigenetics lecture in the YouTube channel if you want to search for that one, which goes into much more depth on DNA methylation as well as histone modifications, whereas in this session I'm really going to be focusing on histone modifications as anti-cancer agent. You'll also find a more comprehensive proteasome lecture elsewhere on the channel, which goes into much more detail on what proteasomes are and all of the different E3 ligases. In this session we're really going to focus on the activity of one key drug, which is Velcade, but look elsewhere on the channel for the proteasome lecture. So dealing with proteasomes first, what we're going to cover is what these proteasomes are and the importance of this protein ubiquitin, and this will help to explain the crucial role that proteasomes play in degrading proteins within the cell. And then we'll go on to how proteasome inhibitors can be used for very specific tumour cell types in very specific situations where these proteasomes can get effectively overloaded by defective misfolded proteins. So we're going to focus on multiple myeloma in this particular session because the drug that we're talking about, which is Velcade, is used to treat multiple myeloma. But then we'll also get onto inevitably how acquired resistance occurs in response to these drugs. So proteasomes are these organelles, roughly about the size of a ribosome, that sort of size, made up of multiple individual proteins. So each of these little balls here is an individual protein, and there are dozens of those to build up an intact proteasome. And they work by having a substrate, which is our protein of interest, that is going to be degraded. That protein becomes ubiquitinated. Now ubiquitin is a protein in its own right, and the substrate gets tagged with a polyubiquitin chain. That polyubiquitin chain is recognized by a ubiquitin receptor at the top of the proteasome. The ubiquitins are removed from the protein by a deubiquitinating enzyme, and then that protein is threaded down through the proteasome, and the protein is chopped up into short peptides, usually of 3 to 8 amino acids. Typically can be longer. As the protein is threaded down through this proteasome, it passes through 3 distinct enzymatic cleavage sites which cleaves between specific amino acids, and you end up with fragmented peptides coming out of the bottom. So here's our target protein that we want to degrade, and when we want this protein to be degraded, we will ubiquitinate it, and this will be done by a ubiquitin ligase enzyme that will tag on the ubiquitin protein. So ubiquitin's a fairly small protein, 77 amino acids, and that will attach to a lysine on the target protein. What then happens is additional ubiquitins are added for a polyubiquitin chain, and this is tagged at lysine 47, lysine 48, K48 of the ubiquitin. So a K48 polyubiquitin chain is characteristic for a ubiquitin chain signal, which is for degradation by the proteasome. Now it's important we mention that because there are other ubiquitinations and different ways of tagging these chains together which don't go for proteasomal degradation. Now ubiquitin is added by a complicated system of 3 main enzymes: ubiquitin activating enzyme called E1, ubiquitin converting enzyme E2, and then the ubiquitin ligase enzyme E3. And there are loads and loads of E3 ligases. You'll come across one already, and that is MDM2, and that is the specific E3 ligase for P53. So from a cancer therapy point of view, the E1 and E2 are not really that important. Well, they're important, but they're not really targetable at all. All that happens is E1 grabs hold of ubiquitin, binds to E2, which is bound to E3. E1 passes the ubiquitin to E2, and then E3 has to bind to the target protein of interest, and when the target protein of interest binds in here, or here, then the ubiquitin ligase E3 can tag that ubiquitin onto the target protein. So the E3 ubiquitin ligases are the ones that are very relevant because they target a specific protein. So the last step of that process is shown here. Here we've got a protein with a degradation signal, and that degradation signal could be one of many things. Things like P53, it hangs around, it gets phosphorylated, it gets stabilized, and it can become phosphorylated, and when you phosphorylate it, it can expose other sort of sequence motifs on that protein, and one of which can be a degradation signal. So if we imagine that this is P53 that needs to be degraded, what we know about P53 is at the degradation signal site, which is where MDM2 binds. If this is an E3 ligase, that can be MDM2. This can be, in this case, unphosphorylated P53. So if P53 is unphosphorylated, in this region here, this can bind with E3 ligase MDM2. If this is phosphorylated, that would stabilize the P53 and it wouldn't be able to bind to the E3 ligase MDM2. But since it's unphosphorylated, it can bind. E2 currently has the ubiquitin. When it's bound in with E3, E3 can transfer that ubiquitin from E2 onto the target protein. We can repeat that protein several times by E1s coming in and providing more ubiquitin, and that tags, in this case, P53 with a polyubiquitin tag, and that will send P53 to the proteasome for degradation. So here are some key cancer-related E3 ligases. So I've just mentioned MDM2 is the E3 ligase for P53. The E6 protein derived from human papillomavirus does pretty much the same function. SCF is an E3 ligase for cyclin-dependent kinase inhibitors such as P21. So they can be produced in response to P53, but then they can easily be degraded by the proteasome. So itch is another E3 ligase, and that will degrade C-flip. If you want to know what C-flip does, you want to look at the apoptotic signaling lecture, but C-flip will prevent caspase 8 activation. And if you think about a situation where you've got DNA damage, we've covered the ATM, which is part of that DNA damage response, which activates P53. Look back to a previous lecture for that. ATM can activate itch, and therefore we can get rid of C-flip, and C-flip is a blocker of apoptosis. So if you have a cell with DNA damage, you want to promote apoptosis, and getting rid of C-flip is one of the ways that a cell can do that, and this rather convoluted route of DNA damage being detected by ATM. ATM, amongst other things, can phosphorylate itch. That allows itch to degrade C-flip and allow death receptor signaling to occur. And then we've got other apoptosis related targets, so BAX, that you will come across that in your apoptosis signaling lectures, can be degraded by Parkin, which is another E3 ligase, and the caspases themselves in apoptotic signaling can be degraded by XIAP, which is an inhibitor of caspase. So that's the background to what the ubiquitin ligase proteasome system is all about. In terms of for cancer specific activity, we have to look for a particular cancer which is multiple myeloma, and that's where we can find a drug that exploits this system and it works relatively well. So in the cell there are lots of misfolded proteins, and misfolded proteins have to be either refolded or degraded, and in many cases proteins that are misfolded are exported to the proteasome from the endoplasmic reticulum, and they have to be then be degraded. There are certain situations where cells are producing vast amounts of proteins, and one of those situations are plasma cells, which are antibody producing cells, which are B cell derived. And when these cells become mutated, they develop into a cancer called multiple myeloma. Now, one of the key things about multiple myeloma is it's associated with translocations of the immunoglobulin genes. Now, multiple myeloma is derived from the plasma cells, and their job is to produce immunoglobulins. They produce vast amounts of proteins. So firstly they have a high amount of naturally misfolded proteins, because proteins sometimes misfold, and when cells produce vast amounts of proteins they will be more sensitive to proteasome inhibitors. But secondly, multiple myelomas have translocations of the immunoglobulin genes themselves, and that creates versions, proteins from those translocated genes which misfold and cause cell stress. If a cell has vast amounts of misfolded proteins, you get the unfolded protein response. You want to get rid of a lot of that protein via the proteasome, so giving a drug called Velcade, also known as bortezomib, that drug is a direct inhibitor of the proteasome, so all of those misfolded proteins can no longer be degraded by the proteasome. They accumulate within the cell, and that causes severe cell stress and should induce apoptosis. And this is summarised here. Velcade is proteasome inhibitory drug. It's used in multiple myeloma, which is a plasma cell malignancy. Plasma cells produce vast quantities of immunoglobulins. When plasma cells have immunoglobulin translocations, you get excessive amounts of misfolded proteins. Therefore, these cells are uniquely sensitive to proteasome inhibitors, and when you give these cells proteasome inhibitors, the cells undergo apoptosis. Since we're dealing with a targeted therapy, inevitably I get onto the conversation of resistance to that drug, and there are two main mechanisms by which resistance can occur. The bortezomib acts on a specific subunit of the proteasome, PSMB5, and what we find is we can get tumour cells overexpressing this specific subunit, so that will effectively outsaturate the Velcade drug. We can also get specific mutations of this gene so that amino acids in close proximity to the Velcade binding site affect Velcade binding, so that prevents Velcade activity. And this is just shown here with cell lines that are being created to be Velcade resistant. So there's a parent cell line with no mutation, generate a Velcade resistant cell line, and we start to pick up mutations that cause amino acid changes around this region, amino acid 45 to 52. So that's the key region, and that's in a very close proximity to where bortezomib or Velcade binds. The other resistance mechanism in multiple myeloma is sort of the differentiation status of the cells that we are dealing with. And multiple myeloma, the bulk of the cells are CD138 positive, and these are the antibody secreting cells, but there's also a population that are more stem cell like, CD138 negative. They're still cancer cells, they've still got the same mutation, they're just undifferentiated stem cell like, and because they are not quite so antibody secreting, they are far less sensitive to the actions of Velcade. So what you effectively can do is kill off the bulk population with Velcade, but you leave behind these cells, and these cells then can continue to grow. They have a slightly different behaviour, they're more primitive, they're more stem cell like, and less sensitive to Velcade. So you tend to then get outgrowth of CD138 negative cancer cells, but you can easily kill off the CD138 positive cancer cells. And these surviving cells may become quiescent, so opt out of the cell cycle, downregulate cellular processes like protein synthesis, and therefore are, by definition, less sensitive to the proteasome. So that covers this group of drugs called proteasome inhibitors, and Velcade is the main drug that you're likely to come across. So we're now going to move on to targeting epigenetic modifiers, and I'll briefly go over different epigenetic modifications and how we can target those in an anti-cancer setting, but I'll mostly be concentrating on histone modifications, but I'll briefly mention DNA methylation as well. So there are two main epigenetic mechanisms, DNA methylation and histone modifications. DNA methylation doesn't really have a approved mainstream anti-cancer agent drug, but it's worth looking at because DNA methylation is intrinsically linked in with histone modifications for either silencing gene expression or by getting rid of DNA methylation, activating gene expression. So if we think about this sequence here being the effectively the anatomy of a gene, here's the transcribe region with all of the introns and exons, here's the promoter region, and then upstream of that are a load of enhancer regions where proteins will bind to specific DNA sequences, maybe tens of thousands of base pairs away from the promoter region. So we think of a promoter as where RNA polymerase binds and where transcription factors bind, but it's more complicated than that. You have got a group of general transcription factors, which are shown here in sort of pale purple. We have RNA polymerase, which can bind, and that's what we need for transcription to occur. We've got all of these other mediator proteins which may bind to other activators which bind to DNA thousands of base pairs away. So yes, transcription factors bind in the promoter region, but thousands of base pairs away you can have these enhancer regions which encourage this large complex of general transcription factors and other enhancer proteins and specific transcription factors. And these enhancer regions can be very significantly affected by DNA methylation. When I talk about DNA methylation, I'm specifically talking about methylation of cytosines next to guanine. So you get something called a CPG island, which is a C, then a phosphodiester bond, and then a G. And on the opposite strand would be a C, phosphodiester bond, and then a G. Now, CPG island should be very, very common in the genome. You know, if you do the maths, a G should follow a C every now and again. And what we find is if you look in normal DNA, which is random DNA, CPG islands are actually underrepresented. There are far fewer of them than you would expect. Whereas if you look in the promoter region of a gene, they're very CG rich. And this is because CG dinucleotides can be methylated at the cytosines specifically. And the general rule is, if those CPG islands are unmethylated, this allows gene expression. If they are methylated, it tends to silence gene expression. And this just shows why it has to be a CG dinucleotide, because if you just methylated cytosines, the opposite is going to be a G. Gs can't be methylated, and that would not allow these methylation patterns to be heritable upon cell division. You want to be able to pass on this methylation pattern to the daughter cells. However, if you've got a methylated C there and the methylated C there, which is opposite the following G, then you've got methylation here and here. You then perform DNA replication. This is a template strand, this is a template strand. The newly synthesized strand is unmethylated at first, but then the methylation status is copied across so that we end up with the same methylation pattern in the daughter cells as in the original parent cell. So therefore it has to be a C followed by a G to allow this methylation pattern to be copied across. In cancer, we tend to see, certainly in some cancer types, an overexpression of DNA methyltransferase. That's shown by this immunohistochemistry staining in brown here. So in some normal tissue, we can see some cells have got DNA methyltransferase expression, and as that progresses through to a poorly differentiated cancer, we've got overexpression of DNA methyltransferase. What that DNA methyltransferase is doing is tagging lots of cytosines with methyl groups and silencing genes. And it's not just silencing random genes, it tends to be doing it in a gene-specific manner and silencing tumor suppressor genes. So this gene, RASF1A, is a tumor suppressor gene, and what we can look at is the CPG islands in its promoter region, and you see that within cancer, these are all methylated, whereas in normal tissue, this is unmethylated. So what's happening here is the cancer is selectively methylating DNA of tumor suppressor genes to silence that tumor suppressor gene, because that tumor suppressor gene is a burden to growth of that cancer. So to combat oncogenic overexpression of DNA methyltransferases, then a group of DNA methyltransferase inhibitors have been described, very well characterized as 5A as a cytosine, which has been tried particularly in myelodysplastic syndromes and other hematological malignancies. And there's other versions of 5A as a cytosine which is in clinical trials. If you want to find out more about DNA methyltransferase inhibitors, this is a really excellent review on their role, a very recent review looking at a whole range of DNA methyltransferase inhibitors. It covers their mechanism of action as I've just described. and summarises a lot of their uses, both preclinical experiments and clinical uses, particularly in hematological malignancies. Now, the other main class of epigenetic modifying drugs are agents that affect histone modifications. So I'm going to briefly overview what histone modifications are before we then get onto the inhibitors. So just a reminder that histones are the major component of chromatin, so DNA wraps around histone proteins, and about every 200 base pairs or so there is a histone core of proteins, and this is an octamer of histone 2A, 2B, 3 and 4, and there are 2 of each within that histone core. So DNA is wrapping around these histones, and the behaviour of the histone proteins and what tags they've got on them determine how tightly DNA is wrapped around. If it's wrapped tightly around the histone, gene expression is going to be suppressed. If it's much looser, transcription factors can gain access to the DNA, and therefore DNA can potentially be transcribed. And this is shown here. So we have DNA wrapped around the histones tightly. If they can uncoil and release, that will allow transcription factor binding to occur, and then that can lock into place. Whereas if these remain tightly wrapped around the histone core, transcription factors cannot gain entry. So tight histone binding to DNA or tight DNA wrapping around histones will prevent transcription of that DNA. The thing that controls whether the histones are control whether DNA wraps tightly around them are the histone tails that stick out. These are the N-terminal tails of the protein. And we have a group of enzymes called histone acetyltransferase which add acetyl groups onto these tails. We can have methylated histones caused by histone methyltransferase, and then we can have histone deacetylases which remove acetyl groups, and then histone demethylating enzyme that remove the methyl groups from histones. And to summarise, all acetyl groups added to histones are pro-transcriptional. So if we have histones that are unmodified, if we acetylate them fully, then this promotes transcription, whereas if we remove acetyl groups, we then make space for the methyl groups to be added, and methyl groups are generally associated with silencing of chromatin. When you get methyl groups, you then get recruitment of heterochromatin proteins which bind and basically very tightly compact the chromatin, preventing transcription factors from binding. So you've got your histone acetyltransferases putting on acetyl groups, promoting transcription, histone deacetylases removing acetyl groups, and then histone methyltransferases putting on methyl groups at specific sites on those histones, and then the ones that are not shown as the demethylating enzymes which reverse this process. There's a unique language associated with histone modifications, and this is summarised here. We've got the amino acid or the relevant amino acids of the histone H3. So histone H3 is the main one which is modified and causes epigenetic modification to gene expression. And what's shown is all the different modification sites that are possible on histone H3. And there's some really important ones to point out, which is K, which is lysine. So lysine 9 can be methylated or acetylated. Lysine 14 can be methylated or acetylated. Lysine 27 can be methylated or acetylated. And lysine 36 can be methylated, as can lysine 4. So lysine 9 methylation forms heterochromatin and gene silencing. Lysine 9 acetylation, in contrast, promotes gene expression. We've also got lysine 4 methylation. This is slightly unusual in that it promotes gene expression as well. That's the only methylation you'll come across which promotes gene expression. Then we've got acetylation at lysine 14 which drives gene expression. Lysine 27 methylation is fundamentally important for silencing of Hox genes, which are fundamental developmental genes, and also X chromosome inactivation, where you get lionization of the X chromosome in females. So we can then get codes such as H3, K27, ME3, and that would mean histone H3 lysine 27, and Me3 would mean it's trimethylated at lysine 27. And this is just an image of the R group of lysine. It's got an NH3 plus group on there, and each of those hydrogens can be replaced with a methyl group as shown here. So this is lysine trimethylated, or you can just acetylate. So on this slide we've got a link to the really good review on epigenetic modifiers in cancer, and this review covers both histone modifications and DNA modifications. And if you want to chase up this particular article, that's the article. So Schiffman et al. And that's the full reference for you there. So it's really well worth chasing up to get a fuller understanding of this topic. But there are two main groups of drugs which are either being used or being trialled as anti-cancer therapies, and those are the HDAC inhibitors, which are the histone deacetylase inhibitors. And the way they work is to inhibit deacetylation of lysine 9 and lysine 27. So if histones become acetylated, they become transcriptionally active. What tumours tend to do at the location of tumour suppressor genes is silence that DNA. Now if we use a histone deacetylase inhibitor, that will encourage DNA to be more likely to be acetylated and therefore cannot be silenced. So by using vorinostat, valproic acid and other HDAC inhibitors, what tends to happen is you add these drugs to tumour cells, and the tumour cells start to re-express epigenetically silenced tumour suppressor genes. So if the tumour starts to re-express silenced tumour suppressor genes, like BAX, then it will suddenly find itself starting to undergo apoptosis, or it might re-express P21 and stop proliferating. So although this is some of the other HDAC inhibitors have much more complicated methods, this is sort of a general mechanism of how some of these might be acting. Similarly there are some of the histone methyltransferases, such as EZH2 and G9A. These two enzymes add on methyl groups, so silence DNA, or silence chromatin, and in cancers you tend to get lots of methylated histones at lysine 27 around the location of tumour suppressor genes. So what we can do is add inhibitors of EZH2 or G9A, and they can then prevent that methylation of histones at those specific sites. What we find in cancer is EZH2 in particular is a histone methyltransferase which is quite frequently overexpressed. So the key point you need to understand here is that both of these histone-targeted epigenetic modifications are predicted to promote re-expression of silenced tumour suppressor genes. Both the HDAC inhibitors and the EZH2 and G9A inhibitors will cause re-expression of silenced tumour suppressor genes. Now the histones are going to be modified, but what we find is that DNA methyltransferase also hangs around in the same complex as HDAC and EZH2 and G9A. So they're all working together, and if you get silencing of the chromatin at the histone level, you'll also get silencing of that DNA at the DNA level. And this is just a screenshot from that review that I showed you earlier, the Schiffman paper, and it identifies the main groups of histone deacetylase inhibitors, the ones that you'll see mostly in experimental therapies. Vorinostat is quite widely used. You see lots of these others used in experimental cell culture and in vivo animal studies. On the EZH2 inhibition side of things, there is GSK343, which is looking very interesting as an EZH2 and possibly G9A inhibitor. So a lot of these are experimental drugs that are not yet in clinical practice, but they are certainly being trialled, and some of them are going through clinical trials, some of them are slightly more routinely used. But have a look through this review and you'll get a really good indication of what is being used routinely and what is just an experimental drug. You have to tilt your head sideways to read this, but what you can see is there are lots of these drugs, particularly verinostat, is being used, rather than being used as a single agent, being used with other agents in combination therapy, and you can see there are lots of clinical trials where this has been attempted. So that's a summary of both DNA methyltransferase inhibitors, how they work, what they do, and how they can be used therapeutically, and the same for histone modifications.